Welcome to week 7. So this week we are focused on gram-positive bacilli, gram-positive rods. So we will kind of go through a few key groups in the gram-positive rods area, starting with the group Bacillus. So Bacillus are aerobic gram-positive rods that are spore forming. So this is going to be the only aerobic group of gram-positive rods that are spore forming that we will discuss later. In the second part of microbiology, we will learn about some anaerobic gram-positive rods that form spores, but this is the only aerobic group that we are going to learn that does form spores, gram-positive rods. So gram-positive rods are similar to gram-positive coxine that our next test is still catalase. We still perform the catalase test. So I always just think of catalase as a great test to perform on gram-positive anything. So Bacillus are very much catalase positive as a group. From there, the testing gets a little different. We have to consider sources, like what source is this coming from? Where's the infection occurring? What does it look like under the microscope? You know, things like that. So that kind of, the testing isn't so flow charted, you know, like I don't have a next step always, depending on what group we're discussing. But catalase is for sure always the first step after gram-positive anything. So in the case of gram-positive rods, we still catalase test it and be catalase positive for Bacillus. Now, our big bad boy out of the Bacillus group is Bacillus anthracis. So that second word should sound familiar. It should sound a lot like anthrax, which is what causes anthrax, is Bacillus anthracis. There are other species of Bacillus besides anthracis. They are more low virulent. There will be one Bacillus cereus that I will ask you to know, and we will discuss as well. So Bacillus anthracis does inhabit the soil, and you can acquire it through inhalation of those spores from the soil. So Bacillus is beautiful under the microscope. They're very large gram-positive rods. A lot of times they are nicknamed boxcar shapes because they're so big. So if you think of boxcars like on a train, each of those is a boxcar. So that's kind of what they are nicknamed after, because they're just super large, super easy to tell that they are a rod shape. There's no question about it when you see it. And then you'll notice here, now this isn't always the case, that there is a lighter colored area in each of these rods, and that's the spore forming part of the rod. Sometimes that doesn't always show through. You might just see them completely dark like these ones here. Other times you might see where that spore forming area is. So at any rate, if you see very big pus rods like this that are growing aerobically, Bacillus should be one of the first things in your brain. So Bacillus anthracis, so like I said, this is anthrax. It is nicknamed wool sorter's disease. So because Bacillus anthracis is found in the soil, it got the name wool sorters because as animals graze the soil, like sheep, they would pick up these spores and they kind of be embedded in their wool. And so when they would, you know, I don't know how often it occurs, but I think it's yearly that sheep get sheared, their wool gets sheared. And those people that are handling that wool can either inhale the spores or they get it through a cut on their skin. And then they have the Bacillus anthracis infection. So that's where it got the nickname wool sorter's disease, is because it could be found, those spores in that wool, from the animals grazing the soil, and they picked it up into their wool, and then the person handling the wool gets it that way. So there are actually 3 different types of Bacillus anthracis infection. You can have a cutaneous type, a GI type, or the respiratory type, which is the big bioterrorism scare type. So cutaneous, again, spores entering through the abrasions, any opening on the skin as they are handling that infected wool or whatever it is that they're handling. So it'll start as kind of like a raised itchy bump, resembling an insect bite, but then within a couple of days, it's becoming a pretty big ulcer, skin ulcer. And then it will kind of have like this classic black necrotic or dying area in the center of that ulcer. The gastrointestinal, which is down here at the bottom, that is just like you might think by ingesting contaminated meat. So if you are ingesting contaminated meat, that will give you anthrax. And then inhalation, again, the respiratory version, which is the top part here, that is breathing in the spores. So again, initially might seem like a common cold, and then after several days, it progresses to severe breathing problems, shock, and it can become septic quickly from that point on. And then that's kind of the premise on how they use it in the bioterrorism tax, right, is that they include these spores in some delivery system that people are going to inhale. And because that is spread the quickest, causes the most damage to people, makes them the most sick, that is why it's a preference. Anytime they look at using bacteria, viruses, whatever it might be, as a bioterrorism weapon, they're always considering how fast is this going to spread? How sick can the people get? You know, that's what they're looking at. And so this is if by delivering it via respiratory system, that is the surest way to get the most sick and to easily target a mass group of people. So that's kind of why they considered this a good one. All right, the other one that I wanted you to know, the other species out of Bacillus was Bacillus cereus. Two different infections here. Seems really crazy, the difference in them, but known to cause food poisoning. That's probably the more common one we know of with Bacillus cereus, and then also eye infections. So food poisoning, if again, those spores are left behind or toxins in that food, you can get sick with Bacillus cereus. Same with eye infections, is anytime you get spores introduced that way. So same reservoir though for Bacillus cereus, much like Bacillus anthracis, definitely in the soil, but again, also going to be water and plants, digestive tracts of animals. And then again, if you're eating that animal, it can come that way. Okay, so media, so augurs that we would grow bacillus on. Definitely sheep blood agar grows everything pretty much for the most part. Chocolate agar grows definitely everything, including fastidious. Thioglycolate broth, another really good supportive broth. So all three of those are going to pretty much grow almost anything. And then again, because it's a gram-positive rod, it's not going to grow on MacConkey, so it will have no growth on MacConkey. That's only for gram-negative rods. You can see some bacillus growing on PEA, once in a while CNA. They tend to grow gram-positives. CNA is more gram-pos cocci based, but it might pop up on there. You know, it's not perfect. All right, so the colony morphology is also very key with bacillus. If you were to see the colony morphology on bacillus, they're very large and they have some unique shape to them. And then if you saw that they're big boxcar gram-pos rods under the microscope, you'd right away be concerned with what you have. So Bacillus cereus are described as really large, gray, spreading colonies. Bacillus anthracis, which is what this picture is right here, is Bacillus anthracis, are also large gray, but they are called Medusa head or comma-shaped colonies. So they have projections. So you can think of comma projections. So if you think of a comma, like right here in your sentence, that's what it means by comma projection. So they'll have like these little projections coming off the colonies. They are not perfectly round colonies at all. They definitely have little projections coming off of them that are unique to Bacillus anthracis. The Medusa head nickname comes from Medusa in Greek mythology, who had snake hair. She had snakes for hair. So the little projections like the snakes coming off is why they nicknamed it Medusa head. All right, so identification to separate between the two, both are catalase positive, gram-pos rods. Hemolysis is different. Anthracis is non-hemolytic, so it's a gamma hemolytic, whereas Bacillus cereus is beta hemolytic. Motility testing, Bacillus anthracis is non-modal, whereas Bacillus cereus is modal. It does have a positive motility. So I always like to think of the non over here for anthracis, non-hemolytic, non-modal. So that's kind of how I keep them together in my head. And then you could put a penicillin disc, like little penicillin antibiotic disc, Stricotolon on your agar plate of organism, put a disc there, reincubate it, and look for susceptibility. Bacillus anthracis is susceptible to the penicillin disc, so it'll have a zone around it, whereas Bacillus cereus is resistant to that penicillin disc. Again, that would be an extra step. There's faster ways to test now, especially if you're concerned with Bacillus anthracis. We have faster ways to test for things, but these are just classic test findings. So the way I remember this is, it sounds really stupid when I say it, but it helps me, is that I think of anthrax as this big bad disease. We've all heard of the bioterrorism. It's been featured in I know Criminal Minds had an episode on it, all these crime shows and movies and stuff. Like you think of this bioterrorism anthrax, and I'm like, but yet it's susceptible to penicillin, the basic of basic antibiotics. So that's how I think of it in my brain, and I remember the results. And then both are lecithinase positive. So lecithinase is an enzyme that they can produce. All right, so those are the basics test that can help begin to differentiate between the two. So a little bit more on lecithinase testing. So this is performed on an egg yolk agar. That's just an enriched agar that will pretty much grow almost anything. So lecithin is a normal component of that egg yolk. So if the bacteria has that lecithinase enzyme, which Bacillus does, it will split that lecithin in the egg yolk agar and make it become a white precipitate, showing a positive result. So down here is the egg yolk agar. They streaked out the organism that they were suspecting. So they put a streak across the agar, incubated it, looked at it, and then there was very much this white precipitate look, and that's a positive for lecithinase. So it's just saying, yes, this bacteria has lecithinase enzyme. It formed that white precipitate. So as we saw on the slide earlier, both are lecithinase positive. Okay, so other lab identification, Bacillus anthracis is known to have a capsule around it. So you can see this by a special stain called an India ink stain. That stain is kind of like a brown stain that when you stain it, it will basically cover everything in brown stain except where the capsule is, because it cannot get through into the capsule. So where the capsule is, it kind of like, it's still clear. So everything else is like brown, but the capsulated area is clear because it couldn't penetrate through there. On media, you can see the capsule, detect the capsule with media that has sodium bicarbonate. That's important to remember, just saying. And then you can also put it in an increased carbon dioxide setting to help stimulate that capsule production. So if you kind of put it on media with sodium carbonate and then throw it into an increased CO2 setting, whether it's a candle jar or CO2 incubator, that will help stimulate any capsule production that might be there. And so that's just another way to identify it might be Bacillus anthracis. You can also look for sporulation. So these are spore forming bacteria. So we can induce sporulation with urea agar. So that urea will basically trigger a nutrient response due to the rapid consumption of nitrogen from the urea molecule. Then the bacteria, so basically it's limiting the nutrients available to the bacteria because it's using it all up in the agar. So then the bacteria goes into survival mode, like, hey, we have to survive. We need those nutrients, and they'll start to form spores. So that's how it induces the production of spores. So that's another way that you can look to see if it's truly spore forming, to know it's a Gram-Pos rod spore forming, is by inducing that sporulation with urea agar. So again, that's another key thing to remember. All right, treatment. Again, as we already said, with the testing process, Bacillus anthracis is still susceptible to penicillin, beta-lactams, basically, because penicillin is part of the beta-lactam groups. Bacillus cereus is not. It's resistant to penicillin. It has a beta-lactamase enzyme that breaks down that beta-lactam ring, so those antibiotics don't work for it. You'd have to use other antibiotics. Okay, switching over to two other groups of gram-positive rods, Corynebacteria and Listeria. General characteristics, they are still catalase positive, but they do not form spores. So we still would catalase test them once we saw we have Gram-Pos rods. They are catalase positive as well. Again, non-spore formers. So Corynebacteria and Listeria are both commonly found in the environment. They might be seen as some normal human flora, with the exception of one Corynebacteria would not be normal flora. And they can be associated with animals. So they're kind of found throughout, you know, in a variety of places. That's a lot of areas. So the two main pathogenic ones that we're going to discuss are Listeria monocytogenes and Corynebacterium diphtheriae. Most of the rest are opportunistic, so you don't see them as commonly. There is one, Corynebacterium jejunum, that we'll mention briefly out of those opportunistic ones. But there's a lot more than just these, but the rest are pretty opportunistic. We're going to stay focused on our main pathogens. All right, starting with Listeria monocytogenes, again, found widely in nature, animal, soil, vegetable matter. Sometimes it colonizes our GI tract. If you do ingest it, and you're pregnant, you can definitely pass it from the mom to the fetus, and that puts that fetus at risk, and that would be a concern. So the virulence factor that is associated with Listeria monocytogenes is a lysine that's called Listeria lysine. It basically kills cells, so hemolyzes, cytotoxic kind of things. It kills cells, and that way it can survive against phagocytes. The main diseases that it's linked with is bacteremia, which is just an all over kind of septic type bacterial infection throughout the blood. and meningitis, especially neonatal meningitis, because there is that risk of it being passed from the mom to the fetus. So that is risking neonatal meningitis. They don't really have much of an immune system to survive against it. All right, it can grow on blood agar chocolate, of course, because it's just a grandpa's rod. When it grows, it's smaller white colonies that are very much beta hemolytic, but they might be a small zone of beta hemolysis. That beta hemolysis might not be really big, but a smaller zone of beta hemolysis. So small colonies with a beta hemolytic zone. They are Listeria monocytogenes. Probably the biggest characteristic test for it is motility. It has an amazing ability to be motile, and it has this very tumbling motility that's the only time we use the words tumbling motility that I can think of is for Listeria monocytogenes. The minute I hear tumbling motility, I'm already thinking Listeria monocytogenes, meaning end over end. Think of like somersaulting. If you ever somersaulted as a kid, or tumbling, when you think of gymnasts, they tumble right on the floor. So that's kind of what it means by tumbling motility, is they can, in that motility test, you would do it as a wet mount. So you take a drop, look at it under the microscope, and you should be able to see them turning end over end. And that's a very characteristic finding here. You could also use a motility auger. And in that case, why is this thing here? There. In that case, if you use the motility auger, incubate it and look at it, it will have a very classic umbrella shaped pattern inside that auger. And I think I have an image of that coming up. So hang on. Very much CAMP positive, hip urate positives. Oh, those are two other tests that you can also perform. Those tests, the only times we have mentioned them so far is when we talked about strep A galactiae. So group B strep last week. Was it last week? Yeah, last week. And we had said strep A galactiae. Group B strep was also CAMP positive, hip urate positive. Group B strep also was a beta hemolytic and also caused neonatal meningitis. So there's a lot of similarities between Listeria monocytogenes and strep A galactiae in that they both are known to cause neonatal meningitis. Both are beta hemolytic. Both are CAMP positive, hip urate positive. Now, of course, the key differences are strep A galactiae is a grandpa's cocci, whereas Listeria monocytogenes is a grandpa's rod, and then the catalase test. Strep A galactiae is catalase negative. Listeria monocytogenes is catalase positive. And then, of course, the motility. Listeria is also very motile, whereas strep A galactiae is non-motile. So just kind of keep that in mind. There's similarities, but there's differences. So this is a nice picture of the Listeria rod here. You can see all the little flagella, which is why it makes it so motile, able to really move. All right, and then on this next one, there is a couple of videos you can play so you can see it doing the end over end tumbling. I think they're fun to watch. I'm not going to click them right now because I don't know if it'll really pop up and the sound and all that. So I'm just going to leave them here. You guys can view them. That way you can see the wet mount look of the tumbling motility. And then, like I said, if you put it into an motile auger and incubate and look, you'll see this umbrella pattern. And so right here at the top, it's showing you where the umbrella part comes out. And then this would be like the umbrella handle. So they basically just stabbed it straight down into that auger, threw it in the incubator, looked at it the next day, and then they saw the outgrowth. So that bacteria essentially moves off of that stab line into this umbrella shape like that. And that's a very classic finding for Listeria monocytogenes. Okay, the CAMP test, a little bit more on that. So you basically streak against Staph aureus down the center of a blood agar plate, and then you take your suspected Listeria monocytogenes, so your patient sample, and you streak it right up until right before it meets that Staph aureus. You don't touch the Staph aureus line, it's just right up to it, so perpendicular to it. Incubate, and you look at it the next day, and you're looking for a little zone of beta hemolysis between the streak and the Staph aureus. So in the case of Listeria, it will have more of a small rectangular zone. You do the same thing when you suspect strep A galactiae. That tends to make more of a triangular beta hemolysis zone. So same technique, Staph aureus on the center, streak up your suspected group B strep, and then incubate and look. So typically strep A galactiae makes more of a triangular zone of beta hemolysis as a positive. Listeria tends to make more of a rectangular zone of beta hemolysis as a positive. I know both here show arrows, but you get the gist. So both are considered CAMP positive. And so there is kind of a look. So each of these right here are the CAMP positives showing that beta hemolysis is enhanced right next to the Staph aureus streak. So this was the Staph aureus. These were our suspected organisms. So showing kind of like a positive camp there. And then the hip urate hydrolysis test. So you basically, in order to detect bacteria that can hydrolyze hip urate, basically what it means is when it hydrolyzes hip urate, changes it into glycine and benzoic acid by an enzyme that the bacteria has. That glycine will then be detected by the reagent producing a deep purple color. So when you add the reagent, if it had that hip uricase enzyme to break down hip urate into those two parts, the glycine will then turn a deep purple to say positive. So it will be deep purple here as a positive, otherwise it will remain colorless as a negative. Okay, before I move off, the one thing I should note here, going back up to the beginning of Listeria monocytogenes, is this whole where it's found in transmission via ingestion. So this is the whole reason why a lot of pregnant moms are told to not eat deli meats, soft cheeses, because of the potential for listeriosis. Listeriosis or Listeria monocytogenes can be found in those deli meats and soft cheeses. And so if they eat that, then there's risk that that listeria in those products gets passed through the placenta to that fetus, and then the risk of neonatal meningitis, since that fetus has no immune system really to fight it. So that is the whole premise. If you've ever, maybe you didn't know that. I didn't know that when I was pregnant. I learned it later, but I probably should have known that. Thankfully, I didn't eat anything like that, but it is usually taught to not eat certain deli meats and soft cheeses for that reason there. Okay, switching over, let's find to Corynebacterium diphtheriae now. So again, a lot of Corynebacteria are carried as normal flora throughout our body. Corynebacterium diphtheriae is more of a carrier state. It is not normal flora. It is a big bad pathogen. We do not have it as normal flora in our body. Diphtheria is just as a carrier state if you do have it. And so, as you can tell by the name Corynebacterium diphtheriae, it causes diphtheria, is the disease. So this is part of the DTaP vaccine. D and DTaP is diphtheria. So that is a vaccine that is given to help prevent and the risk of getting diphtheria. Transmission to pass is via respiratory droplets or, of course, direct contact with contaminated objects. All right, so it has a toxin that causes that diphtheria mainly. So it produces a toxin that will form that infection. There are two different forms of diphtheria. There is the respiratory form, which is the pathogenic form that's really hard on the patient. And then there's a cutaneous form as well. So that exotoxin that it produces also helps it destroy host cells like phagocytes, so that way it can avoid being gobbled up, and destroyed. So respiratory diphtheria, sometimes thought of as like a pharyngitis, sore throat, but basically what it really is a membrane that covers the back of your throat. And that membrane can cover the tonsils, the uvula, your palate, and your pharyngeal wall, resulting in a respiratory obstruction. So your inability to breathe correctly, because now you have this big membrane covering the back of your throat. So yeah, you're going to have a really difficult time breathing. So the toxins can also get released into the bloodstream. And then once it goes into the blood, as you can imagine, it goes throughout the body. So now it's not just a respiratory issue. It can also cause other organ damage once it gets into the blood and be very life-threatening. So that's the reason why they made the vaccine. So this is kind of a look at what that membrane looks like back there. You can see it forming. And so you can see that it's covering a huge part of their back of their throat with a very little hole. That's going to really impede their ability to eat, their ability to breathe. So yeah, very significant. And like I said, then you're also risking the toxins from spreading other places. Cutaneous form of diphtheria, non-healing ulcers, and like they're big ulcers. I apologize for not having a trigger warning, but this picture is next. But yeah, it's not just like a little bit of a skin issue. They dig, like an ulcer is like a very huge skin wound. It's eating away kind of at your skin layers there. So it can form that membrane formation on the skin too. And again, the risk is the further in that those toxins get into the skin, the more they can get to the blood and then again be passed body wide. All right, so identification for Cranibacterium diphtheriae. It is a Gram-Pos rod. They previously Listeria, we didn't show you gram stain Listeria. Listeria were just smaller Gram-Pos rods on gram stain. They were not going to look like Bacillus. Bacillus were the big, large boxcar ones. That is unique to Bacillus. Listeria was a small Gram-Pos rod, nothing unique under the gram stain. Cranibacterium has the ability to look more unique under the gram stain. Yes, it's Gram-Pos rods that are typically smaller, but they tend to take different arrangements on that gram stain. They're said to have palisades, meaning they line up in parallels side by side. They can take the resemblance of the letter V or L, so the two rods can kind of form a V look or an L shape. There was an old nickname for them. It's not the greatest nickname nowadays, but they used to be called Chinese letters. I left it there because in case you see that referenced in any older resources, I don't think the boards say this anymore, but maybe they still do. So I did leave it there in case you see that. Again, it's not a great term nowadays. We know that. But that was an older nickname. So I just wanted you to see it referenced. Again, I don't like it. But I'm leaving it in case that there are resources that are still using that as the nickname. All right, so let's take a look at it. So this is a look at the Cranibacterium diphtheriae gram stain. So Gram-Pos rods, you can see kind of longer, skinnier sometimes. They might have like almost like a club look at the ends of them. You know, this one, I don't see any true V or L shapes or anything like that on this one, but you can kind of see they're just kind of a unique look to it, you know. All right. This one would have maybe a little bit more, let's see, you know, they're kind of grouping together a little bit more. They might have some, this right here has a little V shape here up there if you really look. So there might be a little bit of a V or L shape there. So you can kind of see they're grouping up a little bit together. You can see the palisading is when they line up side by side like each other. So that's happening a little bit. It's not picture perfect, you know, like we think of it in our brain as perfectly lined up side by side the whole way across. No, it's just, they do it, but it's not like the whole thing looks like that always. So take it with a grain. All right, augers definitely should have some very selective augers that we can use if we're suspecting diphtheriae. So there are augers that are just specific to help grow this. Cranibacterium is a Gram-Pos rod, so of course it's still going to grow on blood agar, chocolate agar, a little bit on like PEA, that kind of thing. But you also have some specialized augers too that you can use for this. So we have cysteine tellurite, Tinsdale agar, and Leffler agar. Leffler, not hardly used anymore, but again, I still want you to know Leffler agar is a specific agar for Cranibacterium diphtheriae. So I do want you to remember all three of these are selectively looking for diphtheriae. Tinsdale is a really good one. It takes a very unique look on Tinsdale agar. I think, yeah, I will have a picture of that here in a second. All right, so on blood agar, Cranibacterium diphtheriae is small, gray, sometimes medium white. This, you know, there's a few different looks that it can take. Sometimes it can be beta hemolytic. It just depends on what strain you have of it. So it has a couple different looks. On cysteine tellurite agar, it'll show up black or gray colonies. On Tinsdale agar, it has this really unique black colony with a brown halo look. That is very classic finding for C. diphtheriae. So that is something I would remember. So here's the look on that Tinsdale agar. So the black colony with the brown halo around it. So like this one right here shows it's perfect looking there. So that is very characteristic and should be remembered for diphtheriae. All right. So when we think we're looking for diphtheriae, of course, you got to consider the site. You will only consider it clinically relevant if it's coming, if you see it in a blood culture, because blood should never have bacteria in it. So that is normally a sterile site. So if we're isolating it from a sterile site, if it's coming from a respiratory specimen or like maybe a really big skin ulcer specimen, all of those would be clinically relevant isolates that we should completely identify. Also, knowing their vaccine status would be another thing to take into consideration. Otherwise, the problem is there are so many other Cranibacterium species that are opportunistic. They don't cause that much for disease. We call them craniforms. Sometimes you might hear them called diphtheroids. Diphtheroids are not the same as diphtheriae. It's just like a nickname for all the other Cranibacteria species that are normal flora of our body. that you can find that normal flora everywhere. So craniforms is just kind of means club shaped. And as we saw in the gram stain, they do have that kind of club end to it, that look. So that's kind of why they use that nickname. Again, it's standing for all the others out there. So because it's such normal flora, we can see it as contaminants a lot. So you only really are thinking diphtheriae if it's coming from a really clinically relevant site, like a blood culture or a respiratory sample, and they are not vaccinated, or like a really significant wound infection kind of thing. But again, you have to take into account, like it could just be normal flora off the skin too, because they have such a similar look under the gram stain. So to differentiate C. diphtheriae from others, again, seeing that brown halo with black colony on Tinsdale agar, you could also run a urease test, which is a urea agar. You can stab a slant of urea overnight, look at it. And this should be urease negative, where all of the other cranibacteria tend to be maybe urease positive. So that might also help differentiate it. And then the definitive, really to call it diphtheria, you need to prove the toxin that it's producing. And this is typically done at much larger micro facilities, reference laboratories. There probably is better PCR technology nowadays, MALDI-TOF, molecular kind of DNA technology. That's probably what's really being done now. This is, again, we're just talking old school classic ways of identification right here. So in order to really definitively call this out as diphtheria, you want to prove that it's producing that diphtheria toxin. All right, so again, to prevent immunization status, so vaccines. So being part of that DTaP, which is diphtheria, tetanus, pertussis. All three of those are bacterial infections. So the D here is the part for this one. If exposed, you can treat with different antibiotics, and you would want to follow it up and make sure it's clearing up the way it should be clearing up. All right, the other one that I said we might mention is Cranibacterium jejacum. Again, very much normal flora. You can see it in hospitalized patients. It's not very virulent, which means it doesn't cause a lot of disease unless it has the opportunity to, and the person is really immunosuppressed. Then it might be able to be introduced into normally sterile sites. Maybe it's when they put their IV in, if they didn't cleanse the area well enough, something like that, and that person was super immunosuppressed, then it might become a problem. But otherwise, it's pretty low virulence. So when we look at here, gram stain morphology kind of comparison between the Cranibacterium and the Listeria. Again, gram-pos rods, short, branching, V or L palisading, maybe some club-shaped look to them on the Cranibacterias. Listeria, shorter gram-pos rods. Sometimes it might be called a coccobacilli, where it has that in-between shape, that oval look instead of a full rod shape. Appearance on the colony agar. So again, C. diphtheria may be beta hemolytic, not always. It can vary. C. jejacum is non-hemolytic. Listeria monocytogenes is very much beta hemolytic. And then a little helpful flowchart system. So again, if you do a gram stain and it's gram-positive rods and you're saying they look diphtheroidy, again, meaning they have that Cranibacteria kind of look with the club shape or the V or L or the palisades, you can urease test it. If it was growing on Tinsdale agar with that brown halo and it's urease negative, it's diphtheriae. Again, you'd want to prove the toxin. If it's urease positive, then maybe it's something else. So it's just kind of a look at putting that into perspective. All right, some other organisms in the gram-pos rods. We have Erysipelathrix, Gardnerella, and Lactobacillus. So these are catalase negative. So still gram-pos rods, non-spore forming, but catalase negative on these ones. So these are different than the others that we just all went through. All those others were catalase positive at this point. All right, so we only have 3 organisms out of this group to remember. We have Erysipelathrix ruseopathiae, Gardnerella vaginalis, and then just the Lactobacillus species in general. So starting with Erysipelathrix ruseopathiae, very much carried by and found in animals. So it definitely causes diseases in animals. It is not a part of our normal flora. If we pick this up, we pick it up because we handled something to do with animals. Maybe you're handling animals themselves and it got in through a skin, like a skin opening, a cut, an abrasion. Maybe you are handling like the carcass of animals, animal parts, something like that. And so it's passing through a wound in your skin with that exposure. Gardnerella vaginalis, as the second word implies, it is actually normal vaginal flora. Motor transmission is just anytime that it's allowed to kind of take over. If there's a clearing of other flora and this has more opportunity to spread, it can do that. So, and then lactobacillus, lacto, if you think like milk, very much like your yogurts, different foods. You can find it also normal flora in our GI and genital tract. Very, very rare to cause infection, extremely rare. A lot of times we see this just as contaminants on an agar plate when we see it in the lab. All right, all three of these are low virulence as a result. So they don't cause a ton of problems, but they can. So Erysipelotheryx rhusiopathiae, because that's linked with animals going into the skin through wound opening, it will cause skin infection, what we call an erysipeloid. They name it for what the organism is. So erysipeloids are a type of skin wound infection. So just like how it's passed is where it's causing disease. If it can get in further past the skin, there is always the potential for it to get into the blood and then cause a bacteremia. Gardnerella vaginalis. Oh, I should say on the Erysipelotheryx, for some reason, like I said, it's heavily to do with animals. But when I first learned it back in school, this sticks out in my brain so much. I really learned it heavily associated with pigs, especially. It is associated with more than just pigs, but pigs were a big one for it. Okay, moving on. Gardnerella vaginalis, the infection that's known to cause is mainly bacterial vaginosis. Like I said, it's normal flora of your female genital tract. If it has the ability to cause infection and spread, it can cause a bacterial vaginosis infection. So, sorry, my dogs are fighting over a toy. All right, it can also do UTIs too. So, I mean, kind of the same area if you think of it. Lactobacillus, like I said, pretty much just contaminant. It would have to be an extremely immunosuppressed patient and be introduced somehow into their blood to get sick. All right, media, again, sheep blood, chocolate, CNAPA kind of thing can be found because they're grandpa's rods. There is a special agar called the tween agar that can be used. That's used for a lot of yeast stuff, but also can grow Gardnerella vaginalis as well. All right. So Erysipelotheryx, a little bit more on this. This one has a unique result here in that it is the only grandpa's rod, non-spore forming grandpa's rod that produces H2S, hydrogen sulfide. So that is a black precipitate, kind of a black gas, if you think of it, and they'll make a black precipitate when it's produced. So this is very unique in that it is the only grandpa's rod that's forming that H2S production. So H2S positive for Erysipelotheryx rhusiopathiae here. So that is a unique characteristic to remember. For Gardnerella identification, you can definitely, somebody would visit their provider. Obviously they'd say, I think I have a vaginal infection, and they're going to do swabs and send those swabs. And we're looking to see, do they have a yeast that's causing the vaginal infection? Do they have maybe Gardnerella vaginalis that's causing the infection? There's a few things that we look for, right? So when we get those swabs in, you can do a wet mount. Basically, you can roll that swab on a microscope slide, put saline drop on it, roll it in there kind of, and look at it under a microscope. And with Gardnerella vaginalis, you'll see what we call clue cells. You might have heard of these if you've taken your analysis class, or you will hear them in your analysis as well, because you can see clue cells in both a vaginal swab wet mount and on urine, if it's in their urine, like if they have a UTI with it. So the clue cells are basically your squamous epithelial cells filled with the Gardnerella bacteria. So again, Gardnerella is a grandpa's rod. So you're going to see these large epithelial cells filled with the rods in them. They it's hard to see that they're rod shaped necessarily, but you'll just see it looks loaded, and like the borders of the cell get really obscured kind of looking. And I have a picture here of it. So you get that rough membrane because the bacteria is just filling it up. So these squamous epithelial cells in general are very large cells. They got a singular little nucleus, round nucleus there. Normally this would all be clear. This cytoplasm area would be clear. But when they have these clue cells, they are filled with the bacteria inside. So that's kind of what makes it look so busy in here, is that bacteria filling it up. Otherwise it would have been clear looking. So you can very much find these in urine and in vaginal swabs. They are always relevant when you find them, and you would report them because that's a clear indicator that they have a bacterial vaginosis infection. So again, that is something that we're looking for whenever we get, you know, a swab down. All right, some other Gram-Pos rods. These are considered branching Gram-Pos rods. So these are, again, all Gram-Pos rods that form branching forms. They may form on the auger surface, extend into the air. They are considered aerobic, facultative anaerobes, meaning they can be either or anaerobic. They kind of have a few different growth characteristics that way. Gram stain, very pretty, kind of cool looking Gram-Pos rods. They're really long, chainy look there. All right, so for colony morphology, there is one that has a very characteristic look called Actinomyces israeli, or Israeli, however you want to say it there. This is a part of the Gram-Pos rods that branch are the Actinomyces group, but there's one form on the auger plate that it has these molar tooth-shaped colonies. So if you look at these, they kind of look like if you had somebody that lost a tooth and you were to look at the bottom of the tooth, that's kind of what it looks like, right? A molar. That is a very characteristic look for actinomyces here. So just a note there. Otherwise, they're not that exciting. Besides that, they form the branching and the filaments into the air. This one just also has the benefit of kind of looking molary. Molary. I don't think that's a word. But anyway. All right. So the actinomyces is a group out of this that are part of this group. Not frequently seen in the clinical lab, but when we do, they are known to cause serious human disease. They can be difficult to recognize and to isolate, so they're not fun to play with, in other words. There are two different groups of these actinomyces. Those that are acid-fast or partially acid-fast, meaning they have cell walls that contain mycolic acid in them, and they can stain partially acid-fast positive. And then those that are not. They do not contain the mycolic acid, so they're not acid-fast. So for the acid-fast grouping, we have Nocardia, Rhodococcus, Gordania, Sucamorella, and there is Crinibacterium here. Ignore that. I should have cut that off. I should not leave that there. You should just ignore that part. Do not look at that. Let me just, boop, there, gone. All right, the biggest one probably that I think I discussed the most out of here is Nocardia, but you should just recognize the others anyway. But for our testing purposes, we're going to leave it at Nocardia out of that group. That is definitely acid-fast or partially acid-fast. And then the others is Streptomyces, Actinomedura, Dermatophilus. Oh, these are hard to say. Nocardiopsis. The biggest one on this is I stay focused on Streptomyces. I don't really test much on the others, but they're there mentioned. Okay, starting with Nocardia. So Gram-Pos rod, known to branch, acid-fast variable. So we just say partially acid-fast. Catalase positive, and it is a strict aerobe. It has to have oxygen in order to grow. So this is showing the partial acid fast. Again, acid fast positive will have a purpley look to it. So that is showing that for Nocardia. Here it is on the auger plate. So this is what is meant by the branching, able to form filaments in the air. So you can see that. It's hard to see because this is a top-down view. So on the top-down view, but you can still see, I think you can appreciate that these are rising up into the air. They're not perfectly flat to the auger alone. So hopefully you can kind of get that appreciation. For inhabitants of soil and water is mainly worked on. One second. Okay. I love when my dogs decide this is the time they want to be vocal. I'm so sorry. All right, so Nocardia is inhabiting soil and water. Mode of transmission, of course, is any exposure to that and then inhaling it or through a skin opening kind of thing. So similar to a lot of other stuff. For virulence, ability to resist phagocytosis, they produce large amounts of hemolysins, so like that can lyse cells, things like that. As far as disease goes for Nocardia, they are heavily linked to skin infections, but also the pulmonary. So if they were to inhale it, so respiratory pulmonary infection there. And then again, if it does become disseminated, meaning throughout more of the body, that's a very poor prognosis then for that patient. The other one that I said we would talk about was the Streptomyces. That is non-acid fast. So again, Gram-Pos rod branching, but non-acid fast, found in soil as well. Again, same kind of transmission. Disease it causes is named actinomycetoma, which are lesions of the skin. So if you think of it, all these branching ones very much can lead to different skin infections. similar transmission routes, similar reservoirs where they're found. So they have a lot of similarities that way. So here's a look at the kind of lesions that this is for actinomycetoma caused by streptomyces. All right, again, identification is seeing the gram stain morphology, seeing the way they look on the agar plate, those filaments in the air kind of already starts you thinking. You could do an acid fast to determine if you have partially acid fast positive or not. There's also, again, looking for that areal hyphae. There's, ways to do that. You can also grow in what we call lysozyme broth. I'm not going to test you on that lysozyme broth. That's just a mention. I will not get worried about that or focus on that. So, and then here's maybe a helpful flow chart. Like I said, they're not as crystal clear here testing wise. Just depends on what you think you're looking at. So you get a gram-pos rod. First, you're like spore former or non-spore former. Only on the spore forming side for what we've discussed is bacillus. It can be separated by hemolysis, motility, penicillin susceptibility or resistance. All three of those help separate between them, between Bacillus anthracis or cereus and others. If you're on the non-spore forming side, now you got a bunch more. If they're branching, then you're looking at just the ones we just discussed, Nocardia, Streptomyces, those actinomyces groups in general, and you could test then for acid fast positivity or not, and then go from there. If they're not branching, then you got the rest. Now you can look at maybe catalase to separate out. Again, Cranibacteria, Listeria, catalase positive, Erysipelothex, Gardnerella, Lactobacillus are negative. H2S production over there is just for your Erysipelothex, not the rest. On the Cranibacteria, Listeria side, you can look at motility, that Tinsdale agar, urease, there's a few things there. So it's not a beautiful ability to flow chart. You really have to take in the gram stain look of the rod, because again, they each kind of look a little different as rods under there. And then the agar and just the source of where it came from as well to all help. So that's kind of a mention there. Again, knowing, even though they put the catalase down toward the bottom here, still knowing the catalase results on every single one of these is helpful. So like the bacillus are catalase positive, Cranibacteria, Listeria, catalase positive. And then again, we had mentioned like Nocardia was catalase positive. Where did that go? I know I had it at some point. There, right there. So keep the catalase testing in mind, even though on that flow chart, it didn't have it at the beginning. I still want you to always think catalase test as soon as you have a gram-pos anything. So, all right, we'll leave it there. It's kind of a hodgepodging of gram-pos rods, but we try to stay focused on the most important ones. There's a lot that exist, but a lot of them are low virulence, just opportunistic. All right. Thank you again for putting up with my lovely dogs. I decided to participate with this lecture. Hopefully they weren't too bad on the recording, but we will see you next week.